Andrea Haase. She will talk about the proteomic approach to detect protein modification on cellular proteins after nanoparticle treatment.
Okay, so first of all, I would like to thank the organizers for giving me the opportunity to present our work here today. And yeah, let's start. We have chosen a special model system of nanoparticles to do our first studies. Meanwhile, we also study other types of nanoparticles. And here you see our model nanoparticles. It's silver nanoparticles, which are covalently coated by a small peptide. Here you see the peptide sequence. It's based on cysteine, lysine, lysine. Here you see a disulfide bridge between two of those peptides, and during the synthesis of the particles, this disulfide bridge is broken up, and on the surface of the nanoparticles you will find this 3-peptide. Why have we chosen to use this special type of silver nanoparticles? Well, because the nanoparticles that you end up with are very, very well controlled. So you have a very tight control of both size and shape. You obtain very homogeneous nanoparticles which have excellent colloidal stability properties. And the synthesis is very reproducible. You can produce high amounts, so you have a lot of advantages in the system. We did an extensive characterization of these nanoparticles. I have no time to go in all this detail. It's published in several publications. This is a publication where our cooperation partners from the material side, they describe the synthesis and the material characterization. We have used a TEM, for instance, and here you see the 20 nanometer silver particles that we have used in our studies. For comparison reason, we decided to include a larger version, which is 40 nanometer in size with the same coating, and we included a control silver nanoparticle with the same mean size of 20 nanometer, but with a citrate coating. And as a control particle, we furthermore applied a gold nanoparticle, also 20 nanometer in size, with also the peptide coating. And here you see the difference between a conventional synthesis of citrate-coated silver nanoparticles and the peptide-coated ones. Those ones are much more homogeneous. The polydispersity index is much lower. Our cellular model system was a THP1 macrophage cell system, because we thought to have a macrophage cell system, and the first step would be a very useful cell system to study. And we did a lot of studies before we started with the results I'm going to present now, but I have no time to show all this cytotoxicity and so on and so on. Here you see that we analyzed the uptake of these silver nanoparticles and THP1 macrophages, and virtually all of the macrophages take up these nanoparticles very efficiently and very fast. Interestingly, nearly each cell has between 3 to 5 of these large aggregates of nanoparticles, but also you find a lot of individual nanoparticles in the cell. They are freely dispersed in the cytosol, in the nucleus, even in the nucleolus, and you also find them in the lysosomes. Well, we were interested in the cellular consequences of the silver nanoparticle treatment, and we went for oxidative stress response, as this would be an obvious outcome. And indeed, for instance, we find HO1 induction in these cells. So here you see the difference between treatment with the bigger nanoparticles, 40 nanometer in size, and the smaller particles, 20 nanometer in size, compared to the citrate-coated ones and compared to the gold. You see treatment of gold nanoparticles hardly induce any kind of oxidative stress compared to the control situation, while the treatment with the peptide-coated ones does induce HO1 induction and does induce a protein carbonylation, which is shown below here. The citrate-coated ones hardly induce any oxidative stress as well, and also those ones do not really induce a large cytotoxicity. And the doses given here are mass-based doses, but we also calculated other doses for the publication, of course. So why were we interested in the protein carbonyl formation? Well, oxidative stress-based modification on proteins occur very often in cells, and these are part of the normal redox regulation you find in cells. And the carbonyls belong to one of the most modifications that you can find. It's a stable modification, it's irreversible, and typically it is associated with an impairment or loss of function of those proteins, and even the cell function as impaired later on. You can detect it in a very sensitive manner, and the formation of the carbonyls is a selective process. It's not that just basically every protein is modified, that is just across in the cell, and it's not that just the most abundant proteins get modified most. So you can get this modification either by a direct oxidation of amino acid side chains. Proline, arginine, lysine, and threonine are prone to this, or you can get it via secondary reaction of primary oxidation products from lipid peroxidation or sugar peroxidation towards other side chains. And here you see a more detailed analysis of the carbonyl pattern done with a two-dimensional GEAL approach. You see in this panel, it's the control cells which have not been treated by nanoparticles, and you see a background. Some of the proteins are obviously modified. If you treat the cells with gold, you see those ones who already are modified in the control cells pick up a little bit more intensively, but no new ones appear. While if you treat the cells with nano-silver, you see a massive increase in protein carbonylation, and a lot of new protein spots appear. Well, next we identified the modified proteins by mass spectrometry, and what do you find here? You see a lot of cytoskeleton proteins are modified, actin, tubulin, several intermediate filament proteins. You find a lot of heat shock proteins and chaperones that are modified, so different ones. And you find a lot of proteins of basic cellular pathways, glycolytic enzyme, citric acid, cycle enzyme, pentose phosphate pathway. You find some cell signaling proteins modified. You find some proteins that are involved in DNA replication, transcription, translation that are modified, and just there are a lot of studies available in the literature from other fields who check for protein carbonylation. It's a very well-known endpoint in other fields, and for comparison you find a lot of protein carbonyls in aging process, and here mainly chaperones or cytoskeleton proteins are the main targets, but you also find protein carbonylation in drug-induced cytotoxicity, drug-induced apoptosis, and here a frequent target is glycolytic enzymes. And even some of the diseases are associated with an increase of protein carbonyls, and often, for instance, an increase in the protein carbonylation on the surface of infected cells would be a trigger for an increased phagocytosis of those cells. In some of the disease models, this has been published. Well, to sum up, we have shown cytotoxicity of our model nanoparticles in a dose and time-dependent way. I had no time to go into detail for this. The cytotoxicity was dependent on the particle size, on the coating, and on the material. We have shown uptake by different techniques. The uptake itself is very fast and efficient. The citrate-coated one may be a little less effectively taken up. We find individual particles distributed throughout the cells in cytoplasm and nucleus, but also in lysosomes. We find aggregates, and we think, or we have reason to believe, that different uptake mechanisms do exist. I have not shown any data here, but we are also investigating in this direction. I have shown you data for the oxidative stress development. Actually, also here we have tested for different endpoints, which have a different kinetic. So we find fast oxidative stress responses, some more intermediate time stress responses, and some endpoints which take longer to occur. And actually, some of the effects we see in the cells, such as cytotoxicity, alteration in the lipids, and phagocytotic impairment, are directly connected to oxidative stress. So obviously, in an in vitro situation, the oxidative stress development is a central paradigm for nanoparticle effects. And we believe that the identification of modified proteins, such as protein carbonyls, will help us to understand the toxicity of the different nanoparticles in a more mechanistic and molecular manner. And we will extend those studies in a recently, now it's 1 year, started in a BMBF project called NanoGem, and here we test for more types of nanoparticles in different cell lines, in primary cells and in vivo studies, and you can find information concerning this project on our website. And so I would like to end with the thanks to my co-workers at BFR, but also at BAM at the University of Basel, and at Max Planck Institute in Potsdam. Those are the materials scientists who produce our nanomaterials that were used in those studies. Free University of Berlin that helped us with the TEM studies, and here we get some support for the mass spectrometry. Okay, thank you very much for your attention.
Thank you, Andrea. There are questions from the public?
Just quick question on your image of microscopy image of internalization of nanoparticles.
Yeah.
First, I don't know if you can put the slide, but first, how do you know that what is in the cytoplasm is not ribosomes and are silver nanoparticles? They look the same. And if the aggregation is before or after the internalization?
Sorry. Yeah.
Yeah, the little dots on the back out of the nucleus, how you know there are silver nanoparticles and not ribosomes, for example, which are dark in the microscope? And
Well,
they're free in the cytoplasm. I don't know if you have any chemical analysis or ELSEDAX or...
No, we have not
scanning.
combined this with EDX, but you can clearly distinguish in the image. So we have larger magnification of those pictures, really with a 20,000-fold magnification, and you can be sure it's a homogeneous silver nanoparticle that we see there. Yeah.
Okay. And the aggregates, do you think they are produced before or after internalization?
Yeah, that's an interesting question we ask on ourselves. Different possibilities do exist, of course, as you pointed out. Eventually, maybe most probable that it's aggregation that's occurring in the cell culture medium, and it's a phagocytosed material. But
So there's plenty of endocytosis, and then they will fuse the endosomes?
Also, this might be possible. So currently we are investigating on this. I cannot definitively answer this question.
And regarding the next slide, the microscope images, the peptide wine coated look slightly aggregated, and maybe with very hydrophilic in the hydrophobic TM substrate, but also there is more internalization of the peptide coated. I didn't see the sequence, sorry. And then I wonder if the citrate will get immediately coated by albumin, if it is in the, I don't know if in the cell culture, with maybe more stable, so there is any correlation between colloid, you said there is a perfect colloidal stability, but inoperando. Yeah. Just before, I don't know, because I was just looking at the pictures, it's not very clear. The degree of dispersity on the TM grid is very different from the different conditions. Well, so I don't know if this is any biological impact. It's not a simple question, but...
It's not simple to answer and quick. We have analyzed all this stuff. So, for instance, we have analyzed the aggregation behavior also in cell culture medium over time. Mm-hmm. So we checked after immediately dispersion in the cell culture medium after a few hours, after 4 hours, up to 24, 48 hours. And the aggregation behavior is well, at least for a period of several hours, 8 hours, 12 hours, these peptide-coated ones stay perfectly dispersed. Mm-hmm. After this, aggregation occurs, but as I said, the uptake is very fast. You see them after a few minutes in the macrophages. Okay. Yeah? So, for instance, and if you compare the TEM pictures for the citrate-coated ones, we only see aggregates in the cells. Not that large aggregate, but some small aggregate. So we don't see free nanoparticles for the citrate-coated ones. Only for the peptide-coated ones we see the free nanoparticles.
Okay, thank you.
There is another question in the back.
Are you planning to extend your studies into the carbon materials as well, or do you have any experience or expectations on this front?
On carbon-based materials?
Yeah. Carbon nanotubes, maybe amorphous carbon, no?
No, we have not done anything with those yet, and in the moment we do not plan anything.
Another part of this question is, do you see that same nanomaterials would induce other post-translational modifications, specifically, for example, citrullonation? Is there any information on that front?
As far as I know, there is no information yet available, so that's why we started with the larger study where we also included other material types, so like silica particles, like titanium dioxide, zinc oxide, and so on. And here we hope to answer this question. Yeah, from the chemical field, it's well known. The different chemicals produce a different pattern of carbonylation. And I think you might assume from the nanoparticles, depending on where they go into the cell, whether they go into the nucleus or not, whether they enter the different cellular compartments, lysosomes, not lysosomes, you might expect that maybe the carbonylation pattern might be different. We have to see.
Thanks.
Okay, thanks a lot, Andrea. We have to move forward. So our next speaker